This is Michael Crawford. This is my presentation on cognitive neuroscience and the future of reading. So I want to start off the presentation just really quickly and explain what cognitive neuroscience is. Basically, cognitive neuroscience is the combination of cognitive science and neuroscience. So cognitive science is the theoretical or more conceptual framework or lens through which scientists describe the way that humans think and learn, or in this context, in which they read. It's based in linguistics and psycholinguistics. On the other hand, you have neuroscience, which is more focused on the biological side of cognition. It kind of studies the neurons and the cells within the brain system that affect or enhance one's ability to think or to remember or read. So cognitive neuroscience then is the intersection of these two fields. Scientists study the brain to then understand the cognitive processes like reading. So before we get into how this is done, I just want to give a brief overview into the recency, I guess, of this field of study. So if you look at this graph, which I did pull from Wikipedia, I know it's not the most scholarly of sources, but basically it kind of serves as more of a visual just to show you how recent this topic is. It's rooted way back in the early days by Aristotle, which is similar to Plato, and basically says that the mind is like a muscle. You can see towards probably 200 AD, the brain is first believed to be a source of mental activity, and then you don't hear a lot about it up until the late 1800s. So you can see all that explosion of recent discoveries with cognitive neuroscience. So more specifically, to kind of start to look at the history of it, we break it down into those two frameworks that I was talking about earlier. We have cognitive science and neuroscience, or the conceptual side, and then the biological side. So from the biological side, a couple of important developments are the studies by Broca and Warnke and Angelo Mosso. So Broca and Warnke examined the brain functionality of individuals with injuries and how it affected their language. So they discovered a man who had suffered a stroke, and he could understand language and knew that language existed, but he himself could not speak. And then the second patient that was discussed was this patient who had had a stroke, and he was only able to say the word tan. And so these discoveries were instrumental for the biological side of things because they showed that the brain is directly related to behavioral changes and language capability. On the other hand, towards 1880-ish, you have Angelo Mosso, who was an Italian neurologist. He discovered these bony little defects in his patient's head, and he noticed that they would start to pulse when he gave them stimulating questions or math problems. So this was important because it laid the foundation for the idea that there's blood flow when the brain was being activated. So at the exact same time, in the mid-20th century, you have the behaviorist side of things really starting to pick up. You have Pavlov, Thorndike, Skinner, all with their behaviorist ideas of classical conditioning, connectionism, and operant conditioning. So at the same time that these biological discoveries were being made about the brain, behaviorism is really exploding, and this idea of a stimuli and a response is kind of the main focus of psychological studies. So in the mid-20th century is the first meeting of cognitivists, as they call themselves. So here's where George A. Miller presented his paper. It was called The Magical Number 7 Plus or Minus 2, and it was the real first birthplace of cognitive neuroscience. It's where researchers gathered in one space for the first time to discuss the activity or the relationship between brain activity and these theoretical frameworks. And if you're really interested in that, I would really encourage you to check out the references page and look at this paper that's written in 2003 by George Miller himself, kind of about outlining the timeline of how this all played out. So specifically, when we look at the three major forms of brain imaging technology that we use today, you have PET, which is the injection of radioactive material into the bloodstream, which then goes into the brain. So this is obviously an invasive form of studying the brain, but they can then track where these radioactive materials are going in the brain and then figure out where it's being activated given different stimuli. The fMRI is also not great for children, mostly because patients are required to lie really still in a large metal machine. And what the fMRI does is it measures the charge of water, and there obviously is water in your cells. So it tracks where this charge is being activated given a stimuli. And the last one is ERPs, which stands for event-related potentials. And instead of studying regions of the brain specifically, it measures the timing. So how quickly is your brain responding to a stimuli that is presented? And it's where they hook up electrodes to the brain, and you can see they have all the wires kind of attached to like a putty-like substance on someone's head. So how does this all tie back in? So through these imaging techniques, we learned that there is a large activity. There's a lot of activity happening on the left side of the brain during the brain process or during the brain function of this. So we have occipital, temporal, and parietal lobes, which are the three major lobes associated with print processing and reading. So the temporal occipital lobe develops as reading skill does. The temporal parietal lobe is associated with phonological processing, letter sound recording, and spelling. This graphic, I know it's a little bit blurry, is used to kind of visually show where each of the portions of the brain are being activated. And I mostly included this slide because, as you see, the source in the bottom left is from Shaywitz. And she's quoted multiple times throughout the text in Tracy and Morrow. And then as I started to search for some visual aids, her name was popping up everywhere. And she's basically the source of many studies done in recent cognitive neuroscience, specifically regarding dyslexia. So she's a professor at Yale and created the Shaywitz dyslexia screen. It's basically the leading screen test for determining if K3 children have dyslexia. But you can see in the diagram the occipital temporal lobe in the bottom right over here. It's commonly referred to as the OTC, as well as the parietal temporal lobe are central components of the brain in the reading process. And then the lab school, which is where the source where this image is pulled from, describes that the OTC is often referred to as the letter box. So in this region of the brain, neurons are specifically designed to recognize strings of letters that are put together. So it's kind of interesting, gives a visual. But I wanted to touch back on the dyslexic readers because I think that this is one way, especially in terms of classroom application, that we can really see how this field is important. So when compared to their typically developing peers, individuals with dyslexia showed much lower levels of functionality and growth on the left side of their brain, particularly in those three lobes that were mentioned on the last three. And there's this Simos 2002 study that was discussed in Tracy and Morrow that I thought was important to include because it actually utilized neuroimaging techniques to show pre and post intervention that there was increased functionality within the brain given their intervention. And the last point on here is three major differences. So there's three major differences that the lab school points out between dyslexic readers compared to typically developing peers. So the cortical disorganization, basically through autopsy results, they showed more gray matter in some areas of the brain and less in others in dyslexic learners. So this disrupts ultimately the neural migration and the neuron development. It's not necessarily the same in all brains, but this then leads to differences in activation. So Broca's area, which we learned about Broca earlier, it's often an overly activated region of the brain in people with dyslexia. And so dyslexic readers use this portion of the brain, which is associated with speech, to try and process written words and print processing. So obviously you can see that that would be a major problem. And then in connectivity, the different lobes of the brain are often talking to each other, as you would expect. And in typically developing readers, the left side of the brain and the posterior and the frontal regions, they're all talking to each other. But in dyslexic readers' brains, what will happen is that the information is in the frontal lobe, and instead of taking it to the posterior part of the brain, it will try to take it to the right side of the brain, which then leads to struggles connecting written letters to speech. So the information processing theories I thought were kind of a good way to sum up a theoretical foundation in the idea of cognitive neuroscience. So it's a cognitive theory that describes the processing, the storage, and the retrieval of information processing from the brain. Atkinson and Shiffrin, they have the idea of information processing model, and it's one of the most well-known information processing theories. It has the idea of a stimuli, your perception of the stimuli, then going to short-term memory, to working memory, to long-term memory, and kind of this idea of how humans learn. And then at the same time, the second theory is behaviorism, which was the major line of thinking as cognitive neuroscience research began to pick up steam. And George Miller discussed the importance of behaviorist theories in his 2003 article, and it states that he stated that in order for this field of cognitive neuroscience to take off, they needed to be able to successfully integrate scientific data with these behaviorist observations that were taking place. And then from a very internal sense, you have the connection to physiological theories, which I'll talk about in the second part of our presentation. But basically, it's the concept that neural connections help us read, from identifying words to decoding to speech. If you have a healthy brain from a healthy body, then these repeated neural connections are going to happen more freely and more frequently. So here's a list of references. And again, if you would like, I really encourage you to check out this 2003 article by George Miller. He just kind of talks about the timeline of how this field has taken off and what happened in the mid-20th century to let it grow into the field that it is today.